Right. So I'm just gonna start off by giving you an overview for those of you who don't really know me, don't really know about what I do. I just wanted to give you a little bit of an overview of my appointment, and my appointment is primarily extension, followed by research, including being an assistant professor. I'm also the Medical Entomology Extension Specialist. So that means that it's my job to translate FMEL faculty members as well as other medical entomologists' research results into practical information that can help stakeholders make informed decisions. Also, it's my job to create and provide stakeholder extension programs. And so when we're talking about medical entomology, you know, you might be wondering, who is a stakeholder? Well, we're stakeholders being citizens of Florida where it's hot and we deal with a lot of mosquitoes or other arthropods. Additionally, one of my major stakeholder groups that I work with are mosquito control programs as well as public health professionals. And of course, those stakeholders can change. And additionally, I also provide expertise to the county extension faculty in all the extension offices in the 67 counties within Florida. So for the public, I provide different types of information through different methods, and I have some of the methods listed here. The first method is going to be through EDIS documents, and I'm not going to talk about that because Rebecca is actually going to talk more about that later. Additionally, I also keep everybody up to date on happenings of the lab, new research publications, when faculty, students, postdocs are giving presentations, that kind of awesome stuff on social media. So I created a Facebook page for FMEL and Twitter. We're now up to about 100 Facebook followers and over 150 Twitter followers, so a good start. Additionally, some of the materials I use to inform the public are going to be products that are available in the UF IFAS Extension bookstore, such as this booklet here on integrated mosquito management around homes and neighborhoods. This product is for sale, and it's definitely a product that is useful for mosquito control programs to give out at outreach events so that they're not having to recreate the wheel and create these themselves. Additionally, I provide information through giving TV, radio, and newspaper interviews. I also provide information to the public by giving presentations and trainings. So I give presentations to different groups such as master gardeners. I gave a presentation on bromeliads and mosquitoes to the St. Lucie County Master Gardeners a few months ago. Another group that I work with that I'm really excited to be working with is the Bradford County Buzzbusters, and they're actually a citizen scientist group. The mosquito control program in Bradford County is one person, so so there's no surveillance that could happen within that mosquito program, because really that person, he's not actually full-time mosquito control. He pretty much just applies pesticides. So what some citizens have actually started, this was started long before I came into this position, some citizens actually got together and they work with the extension office in Bradford County, and they actually set up mosquito traps at their homes. And the county is broken up into 12 different sections. And so the people have the mosquito traps at their homes. They identify the mosquitoes as much as possible, as far as they can. Usually it's just going to be to the genus level. And then they provide that information to the extension office. And so that extension office then communicates that information to mosquito control, and then they actually have surveillance that they can use to face whether or not they've reached the threshold for applying pesticides. So I think it's a really awesome program. And so I meet with the buzzbusters on a quarterly basis and provide training on things like mosquito identification, mosquito trapping techniques. And this is also a program that I would love to take and get started in other counties where there are just with only just like one person in mosquito control. And that certainly is the case in quite a few counties, especially in the Panhandle. Also, to get information to the public, I work with the Extension County faculty. If they have a resident that comes into the office, or if the resident emails them and has a question related to medical entomology, they want my input, they can call me, they can email me, and I provide them with information. Additionally, I'm working on Train the Trainer program for the Extension County faculty, where I'm going to be educating them in things like mosquito identification, also integrated pest management, and ultimately the goal is to hopefully reduce mosquito populations. And this is something that I've just started working on, and so far what I've done is provide a presentation on mosquito-borne illness prevention at the Central District Symposium, Extension Symposium earlier this year. And then that presentation was given by multiple extension agents to their clientele, whether it, I think one was a Master Gardener Club or group, and then one was actually for new homeowners. So that's certainly something that is in progress. And of course, if the public has questions, they can contact me directly, and this usually happens through phone calls or emails, and I get a lot of interesting phone calls and emails, as you can potentially guess. So here, someone, they sent photos of a lesion on the foot and asked if a certain type of roach, the Florida woods roach, could cause this type of burn on the foot, this type of wound that looked like a burn. And I told the person, no, I didn't think so. I think it was potentially a brown recluse bite. This person's in the Panhandle, so it's within the potential range. But the person disagreed with me and said, no, it was actually a Florida woods roach. So additionally, I get phone calls and emails and packages from people with delusionary parasitosis. I didn't really realize how common that is until I started in this job. And so I get photos of what people think are insects that are coming out of their body. And that is just all I can do is just look at the samples that they either provide to me or provide the photos of. And actually, I want to spend time and I look for insects. And if I don't find the insects, all I can do is just tell them, I didn't find any insects in your samples. Also, because mosquito control programs are really important stakeholders within medical entomology, I provide 3 different extension programs geared towards mosquito control programs. The first one is to provide expertise, education, and training to improve mosquito control in Florida. And this training is can either be generalized or individualized to meet the specific needs of a mosquito control program. And this extension is going to be done through emails, phone calls, visits to the programs, or they come and visit us at the lab and receive one-on-one training by members of my lab, or there can be both. And then this just gives you an idea of some of the programs that I've worked on an individual basis with closely recently. Also within the same extension program is providing generalized training, and so leading generalized training and workshops, as well as participating in these training and workshops. And one such training that I'm really proud to be part of and helping to plan, and also I teach a course as part of the Florida Mosquito Control Association's Dodge short courses, which is just a yearly training, a week of training. There's different courses that people can register for during that week that are different links, and they are on different topics. And at this point, this training is actually, this training has people that attend from all over the world, and it's really considered one of the best training programs for mosquito control. And so a lot of the programs or the trainings and workshops that I either lead or participate in are going to provide CEU opportunities for public health insecticide applicators. Additionally, another one of my programs is to work directly with the Extension County faculty to allow them to deliver mosquito management training. And that just is in an effort to meet the demand of these programs that, I can't be everywhere, and especially in the Panhandle, they may need training, and I'm not always, cannot always be there. And this is something that is very new. I'm working on it, and I'm currently have 3 different counties in the Panhandle that I am developing this program with, and hopefully we'll be expanding it. And then the final mosquito control extension program I have is specifically just providing results-driven management recommendations based on the resistance testing research that my lab conducts. And this is research that Davi and Anna are going to talk about soon, so you'll get to learn more about that. But these management recommendations that I provide to the control program managers, they're specific to each mosquito population, and usually when I provide the recommendations, I'm going to provide multiple recommendations. Usually I provide the active ingredient that was the most effective at knocking down or killing the mosquitoes. And so that way they know if they are in an arbovirus transmission situation, this active products with this active ingredient are going to successfully knock down and kill those adult mosquitoes and hopefully prevent further arbovirus transmission locally. Also, I provide suggestions to prevent increase in resistance rates. Usually involves ideas about rotating products or potentially increasing the application rate of their product to the highest application rate. And really the goal of that is to allow our existing insecticides to continue to remain effective against our mosquito populations. So I've talked about my extension. For my research, my lab is currently focused on basic and applied research related to insecticide resistance in mosquitoes, as well as novel forms of mosquito control. And this type of research really does help inform my extension, and it actually really allows there to be almost a continuum in my appointment where there's not necessarily a break sometimes between where the extension ends and where the research ends. It all just kind of flows together. And I will say that because I do give trainings and all over the state, I travel, and luckily I have incredible lab members that are able to continue getting the research, keeping the research going while I'm traveling. And I certainly want to thank my lab members for all that they do, and I certainly want to thank my lab manager, Daviela. She's really the glue that keeps my lab together. And so because the research in my lab wouldn't be possible without my lab members, now I actually want to turn it over to them, and they're going to discuss these current and recent projects that have taken place in the lab. But first, Rebecca is going to talk about EDIS documents.
Thank you, Dr. Buckner. Hello, my name is Rebecca Zilmer, and I will be discussing another extension project called EDIS. The Buckner Lab has been working on publications for the UF IFAS Extension EDIS website, and EDIS stands for Everyday Information Source. It is a comprehensive single source archive of current UF IFAS peer-reviewed publications. These publications are free to the public, include over 7,000 publication titles that are accessed over 26 million times annually. It is a product of UF IFAS extension programs, and EDIS is funded through UF IFAS and supported through state legislators. The Buckner Lab has been working diligently to provide new and updated publications to add to the Medical Entomology EDIS pages. The current working topics are primarily vector-borne diseases, including dengue, chikungunya, eastern equine encephalitis, West Nile virus, and several others. Here's an example with information that will be provided in an EDIS document about mosquito vector diseases, including the pathogen, information about the vector, transmission cycle, distribution, symptoms, and prevention. EDIS has become the single source for all electronic extension publications, and it is tailored to allow extension units to print documents on an as-needed basis. Recently, the UF IFAS EDIS publications have brought together other valuable web resources within a framework in a single web user-friendly theme and framework. These EDIS documents are not only peer-reviewed, but they're research and science-based, and we want to make sure that readers are getting accurate information. We want users to come to UF and rely on UF as a primary resource. And again, it's free and accessible for all. Thank you.
Thank you. Hi, my name is Ashley Paige. I've been in Dr. Buckner's lab for maybe 6 months, and I'm going to share with you a little bit about my experience working with SC Johnson. SC Johnson is a leading manufacturer in household products. Many of you might know of deep products like OFF. SC Johnson is a family company working towards a better future. Being involved in this lab has allowed me to help SC Johnson conduct research that they would otherwise have to spend extensive amounts of money and travel time doing. This is because SC Johnson is located in South Carolina, or no, I'm sorry, Wisconsin. Wisconsin. So as you can see here, the travel time for us would be 30 minutes, whereas they have 19 hours. So SC Johnson chose to do the research in Florida for a few reasons. Using some weather guides, I put this chart together to show the variation between rainfall and humidity and heat between Wisconsin and Florida. Understanding that mosquitoes require water and decently high temperatures. That leaves Wisconsin out for half of its year. So being in Florida, working with SC Johnson, it was a real pleasure to be able to help them extend their knowledge on different products that they're making. This is important because mosquitoes all around the world are spreading different types of viruses. The Culex, for example, is known for the West Nile virus, whereas Aedes is known for a variation of chikungunya, dengue, and Zika. And to give a little insight on distribution, this is a map put together from the CDC from 2004, demonstrating where malaria is caused or still ongoing. And this is from the World Health Organization from 2013, representing dengue virus. So by helping SC Johnson conduct annual research on these mosquitoes, we can better prevent these diseases all around the world. While SC Johnson has accomplished so much with a variety of their products, the team I worked with dealt specifically with mosquitoes and the diseases that they can transfer. As we've gone over research in this product, field has great potential in combating mosquito-borne diseases, important in the US as well as outside globally. With research further, SC Johnson can advance repellents and application methods. And finally, to a large concern, this information can offer currently infested areas a disease-free future. And with that, I end my presentation, and I think Ciara takes over. Dabby takes it over.
Hi everyone, I am Daviela Ramirez, and I'm Dr. Eva Buckner's lab manager. And as she mentioned earlier, I will be further discussing the current Florida Department of Health Surveillance and Insecticide Susceptibility Grant we've been taking part in since the beginning of this year, and will be wrapping up around the end of next year. So we're actually building on the similar framework previously instituted in August of 2016 by Dr. Roxanne Connolly and current PhD student Casey Parker. So our current grant has two primary objectives. The first being to conduct surveillance and surveillance data, which is done when we ID the adult mosquitoes from every single site that's received. And the second being to conduct insecticide susceptibility tests by performing the CDC bottle bioassays. And the three species we're targeting are Aedes aegypti and albopictus, along with Culex fasciatus. Performing these bottle bioassays using pyrethroid and organophosphate insecticides will improve our understanding of domestic mosquito control operations, along with also aiding them in managing their insecticide treatment schedule. Additionally, the data obtained from the assays will give us an insight on how to better avoid or manage future arboviral outbreaks. So once we've gotten in contact with the local mosquito control agencies, county extension agencies, and hopefully in the future, the general public, the next step is to send them the supplies needed for collecting the Aedes and Culex eggs. Just to give you an idea, this is what's sent in Aedes kit. So since a lack of funds is a reality for many of these programs interested in taking part in this grant, we've also recently begun supplying prepaid postage slips to those that are interested but may have some sort of setback monetarily. And once Aedes eggs are received, they're counted and stored by county and site and are hatched for future, in the future, whereas Culex rafts are hatched immediately upon receipt. Now, in terms of the mosquito rearing, all Aedes sites are hatched once we've collected at least 100 or more good F0 eggs, and they're ID'd to species and separated if there are Aegypti and albopictus within the same site. All other Aedes species, such as Aedes triceriatus, will just serve as additional surveillance data. While in their bug dorms, adult mosquitoes are able to freely mate, go through several rounds of blood feeding where we collect either F1 or F2 blood-fed eggs. And sites with less than 100 F0 adults commonly require that the F1 adults should be blood fed in order to have enough F2 adults for 6 bottle bioassays. With the Culex, because we receive the rafts and they don't necessarily store the same way that the Aedes eggs do, as I had mentioned in the previous slide, we hatch them as soon as we receive them, and we ID them as larvae when they're in the 3rd or 4th instar stage. And if there aren't enough F0 Culex twinks, which is the species that we're targeting for this specific grant, we go through several rounds of blood feeding with them as well, and we collect their rafts by placing plastic cups with lactalbumin and yeast water. And all additional species, such as like Nigripelpis, are assayed as F0 adults so long as we have enough mosquitoes available for 1 or more assays. If there are not enough adults available, they just serve as additional surveillance data as well. And lastly, onto the topic of resistance testing. Some of you may be asking, what is insecticide resistance? So the CDC defines insecticide resistance as a genetic change in response to selection by toxicants that may impair control in the field. It's extremely important to understand that the CDC bottle bioassay is a tool designed to detect resistance before an active ingredient before that active ingredient is no longer effective. In our lab, we test 2 organophosphates here and 4 pyrethroid insecticides. When it comes to actually performing the bottle bioassay, each assay has 1 control bottle which contains just acetone and 4 treated bottles which contain the specific concentration of active ingredient that we're using for that test. It's also important to know that each treated bottle serves as 1 replicate. So there's 4 replicates within each assay. So as I had mentioned, the control gets coated with acetone, and the 4 treated bottles get coated with the insecticide being used for that current test. Once they've been coated and rolled, they go inside of a room that is full temperature in the dark with the caps off so that the drying process can continue happening until we are ready to use those bottles and introduce adult mosquitoes into them. So once we're ready to go ahead and begin our actual assay, we introduce approximately 25 adult mosquitoes into each bottle, which means you need about 125 total for each active ingredient tested. Throughout the 2-hour period of the bottle bioassay, the number of dead or alive mosquitoes are recorded on a data sheet, and from that data sheet, the susceptibility or resistance status of that specific population is determined by the % of dead field mosquitoes at the diagnostic time. And the diagnostic time is the time it took that same species susceptible strain to reach 100% mortality. And once we have those % mortality at the diagnostic time percentages, then we are able to understand if that population is either resistant, developing resistance, or susceptible to that specific active ingredient. And we also do an additional little step with Aedes aegypti and any Culex species that we run bottle bioassays with. These 2 species are known to exhibit something that's called knockdown resistance to pyrethroid insecticides. And what that essentially means is that when they are being directly exposed to pyrethroid insecticides, they may appear to be dead because the insecticide is only paralyzing them. But once they're removed from that direct exposure, in this case, those 4 treated bottles, there, they are able to come back to life because they're no longer being exposed to that active ingredient. So after the 2-hour assay is done with the Aedes aegypti and Culex species, we transfer them to clean cups that have a sugar cotton and plastic cup over them, and we take an additional 24-hour reading. This 24 hours is determined from the starting point of the assay. And we compared that 24-hour percent mortality to the percent mortality at the diagnostic time. And that is how we determine if they are exhibiting knockdown resistance. So if there's a higher percentage at the 24-hour mark, then they are not exhibiting knockdown resistance because they're still actively dying, even though they've been removed out of that direct exposure. Whereas alternatively, if they decrease, the percentage decreases at the 24 hours, then they are exhibiting knockdown resistance because they're able to get back up once they've been removed from that direct exposure. And now I will introduce Ana Reomero-Weaver to go further into the process of after the assays have been completed.
Good job.
Hello, my name is Ana Reomero-Weaver. I'm a technician in Dr. Buckner lab. And as Davi mentioned, I'm going to tell you about some of the results we have gotten utilizing the Boro bioassay. For this study, well, I would like to mention that the results I'm going to present are for the studies that have been conducted until September of this year. And this slide, this slide shows the population where the mosquitoes were collected. We work with 3 populations of mosquitoes: Aedes aegypti, Aedes albopictus, and Ciara Schloop. And you can see that they are all distributed through the state of Florida. In the next 3 slides, I'm going to show you a comparison of the percent of mortality within each species of mosquitoes. And I would like to emphasize that these results cannot be extrapolated directly to the field. We cannot say, for example, that in the Pinellas County, the Tofenprox exhibit a 10% mortality. These studies have to be analyzed within the same population. And what we can say is, for example, again, in the Pinellas County, that malathion is susceptible. I mean, the population is susceptible to malathion. There is more resistance with Tofenprox and less resistance with nail. In the case of Aedes albopictus, both populations show susceptibility to all the ingredients analyzed. In the case of Ciara, we can see that all the populations show different degrees to the parameters analyzed. And in the case of the Brevard population, there was way less resistance to organophosphate, malathion, and nail. And in the case of Miami-Dade County, the populations were sensitive to malathion and nail. As Davi said, we also have the opportunity with the CDC bioassay to study the mechanism of knockdown resistance. This is an example with the Q squink in Brevard Viera. And as you can see in Tofenprox and dexametry, the percent of mortality at 24 hours decreases comparing with the percent of mortality at the diagnostic time. So what this tells us is, for example, in the case of Tofenprox, that from the 46% that we observed dead at the diagnostic time, only 3% were really dead. The other 43% were knocked out. So once we collect all the data, Dr. Buckner sends a report to the control program manager, which includes all the information of the site that has been analyzed, the species, and then she has included a table with the active ingredients, the concentrations, the diagnostic time, and the results that we obtain. In the case that we observe knockdown resistance, she will add an additional column with those results. And this project is very important because it helps the programs that have no surveillance and no resistant testing. It also helps them to determine which vectors are present in their populations. It also gives information about what active ingredients is better for the adult mosquitoes present in the populations. And it's also very important to prepare in the events of diseases transmitted by arrowbiters, or in the case that outbreak happens, it will there will be more information about what insecticides use. Also, when to prepare for natural disasters, especially in our states that we have the threat of hurricanes. And we have this site called CDC site. It's another resource where we collect where we compile all the data from all the bioassays that have been done under the grant. It not only provides information about mortality, it also provides information about all the conditions in which the studies were performed. And it constitutes the first-hand information about the insecticide resistance, especially in those areas where the CDC may not have a direct contact. This is government authorized access, it's not access to everybody. Thank you, and now Natalie is going to tell us about another important project.
Hi, my name is Natalie, and I will be talking about the Intocare trap project against the Culex quinx. And just to begin, the Intocare trap is designed to be attractive for Aedes aegypti and albopictus females. However, while Dr. Buckner was performing a field evaluation in Southwest Florida, she discovered that Culex were actually attracted to the trap as well, laying their rafts inside it. And so now we are using her semi-field experimental design to actually test that here on campus using our lab-reared Culex fasciatus. So a little bit about the process. First, we are assembling the trap, and the trap is filled with water and installed, and then we install a plastic ring with a gauze strip that is coated in 2 bioactives. And then we set it inside our experimental tent enclosure, and then we release blood-fed females into it, and we also have a negative control screened enclosure. And we also release larvae into each of the flower pots that are surrounding the trap inside the experimental setup, and proceed to pick up the replicate a few days later. This is an example of what our experimental test setting looks like, with the Intocare trap in the middle, and then 4 surrounding flower pot containers where the larval breeding sites are. And then this is an example of the negative control setting with 5 flower pots and no Intocare trap. And the number of gravid females that are released is the same between both tents, as well as the amount of larvae. So through this process, we're seeking to answer 3 questions, and the first being, is the trap attracted to the gravid Culex quinx fasciatus? And this experimental process is ongoing. We have postponed until spring for more suitable weather, and also earlier this fall, we were experiencing a little bit of difficulty with our screened enclosures as adults were escaping. But through a little trial and error, we figured out new materials to use and are receiving better recapture rates of all of our adult females. And then the second research question that we're seeking to answer here is, can the gravid Culex quinx auto-disseminate the pyroproxypin, which is one of the bioactive ingredients inside the trap? And so now we have data from 11 out of 15 of these replicates used in this setting, and we are still collecting the data from this. But this diagram is depicting what happens at the adult female is attracted to the trap and makes contact with the gauze piece inside, proceeding to disseminate, or so we hope. And then the third research question that we're seeking to answer here is, can this fungal spore be, so can this fungal spore exposure reduce survivorship of these adults that we are recapturing. So after all of the adults from the 2 setups are recaptured, they're transferred to clean cups and monitored daily for recordings on this. And the fungal spore is a slow-killing adulticide that has been proven useful in the Aedes aegypti in the field, and so that's also another reason why we're interested in doing this with the quinks. And then, seeing as this experiment is ongoing, we are still collecting results, but so far we have seen that yes, the Intocare trap is attractive to the females as they are flying in, and this is proven with the rafts that we have seen in there. And there's a significantly higher percentage of rafts laid inside the trap versus the 4 surrounding flower pots. And furthermore, that yes, the gravid Culex quinquefasciatus can also auto-disseminate this pyroproxfen larvicide from the trap to the surrounding containers. And this is noticed through the percentage of adult emergence inhibition being higher inside the trap versus the 4 surrounding flower pots. And now Ciara will speak. Thank you.
Hello, my name is Ciara Schloop, and I recently graduated from University of California, Davis. I am grateful to be Dr. Buckner's master's student, and I have just finished my first semester of courses here at UF. I have been working on a rough outline for my research proposal, so I figured today I would give you a brief overview of the concept I'm looking to work on, some questions of interest that I have, and why I think answering these questions can be useful for mosquito control. The major topic of my research will be looking for a relationship between circadian rhythms and metabolic detoxification in mosquitoes. For those of you who don't know, many organisms, including insects, exhibit circadian rhythms that induce behavioral and physiological responses. Daily rhythms can be observed under light-dark cycles, or they can be innately driven, and you can observe them under constant conditions. In mosquitoes, these rhythms are controlled by the joint action of both environmental light and dark cycle, as well as circadian. Functions of the mosquito that are controlled by the circadian rhythms include feeding, mating, oviposition, odorant sensitivity, and metabolism. So now that you have a bit of background, I'm going to go over some of my questions. My first question is metabolic detoxification a resistance mechanism found in permethrin-resistant Florida Aedes aegypti population? So it's kind of a mouthful, and to give you to kind of explain that a little bit, metabolic detoxification is another mechanism of resistance in mosquitoes and actually a lot of other insects that require control of pesticides. Metabolic detoxification itself is basically when certain enzyme-mediated reactions cause the degradation of toxicants. I think that the most widely understood concept of this would be the monarch butterfly and its ability to eat the poisonous milkweed plant. That's because of metabolic detoxification. In this case, the toxicant is a natural toxicant from the plant. In our case, permethrin is a man-made insecticide toxicant, so it's a similar concept of that one. For my second question, are there time of day changes in metabolic gene expression? Another way to say this would be during different times of the day, are there different levels of enzyme expression or activity of our enzyme of interest? And when would this be? Because detoxification responses can't be constitutive, knowing the time of day when the enzyme's activity are at their highest and their lowest in these mosquitoes can allow us to determine if differences in the enzyme activity are enough to confer differences in susceptibility to the specific active ingredient, in my case, permethrin. That leads me to my final question of interest is there a relationship between time of day changes in metabolic gene expression and permethrin susceptibility in these resistant Aedes aegypti populations? If I can find the relationship between susceptibility to this active ingredient and the time of day, this can provide important information for mosquito control when designing and implementing integrated vector management programs. This will hopefully provide some more specified information to the recent resistant maps, for example, the ones we're creating with the FDOH grant that are being made. Thank you. And if you have any questions for any of us, we will take them.